Hello, my name is Adam Hassan, and I am a PhD candidate at McGill University. And today I'll be presenting my talk entitled Cathepsin B, delivered by an attenuated Salmonella Typhimurium vector, confers therapeutic protection in a murine model of chronic schistosomiasis. I'd like to extend a thank you to the organizers of BSI for their invitation. Now, before I start, I'd like to highlight a group of diseases known as neglected tropical diseases, or NTDs, which comprise of infections by protozoa, viruses, bacteria, and helminths. And the current COVID-19 pandemic has caused a partial or total interruption of various health programs, where the global NTDs agenda is not an exception. And this will have severe impacts on the lives of millions of people worldwide in low and middle income countries. And one of these NTDs is schistosomiasis, which is a fresh waterborne disease. Globally, there are over 250 million people infected and approximately 800 million people at risk of infection. And the disease is endemic in subtropical and tropical regions of the Caribbean, South America, Africa, and parts of the Middle East and Asia. Praziquantel is an oral antihelminthic, which is the most effective drug against schistosoma species and results in adult-born paralysis. However, it is ineffective against any other life cycle stage of the parasite and does not confer protection against reinfection, meaning that individuals under drug chemotherapy are susceptible to being reinfected over and over again. And due to the lack of a commercially available vaccine, the WHO has set a low threshold at 40% protection for what they consider to be a good vaccine candidate. There are currently a few anti-schistosome vaccines in clinical development. One of them targets S. hematobium, which is responsible for urinary disease, and the other three target S. mancini, which are responsible for hepatic and intestinal disease. S. mancini will be the species of focus for the rest of this presentation. And all four of these anti-schistosome vaccine candidates are given intramuscularly and comprise of adjuvanted recombinant protein doses, which target surface membrane proteins of the adult form. Comparatively, in our lab, we focus on Cathepsin B, which is a cysteine protease that's located in the gut of the schistosomia in adult worms, and it's a digestive enzyme used to degrade host blood macromolecules. And there have been various protective efficacies when we use Cathepsin B in different formulations, ranging from 54% to up to 90% protection. And my project focuses on Salmonella Typhimurium YS1646 strain as a novel delivery platform for an S-mancini vaccine. It was originally produced as an anti-cancer treatment, and through disruptions of the purine gene, which makes it a purine auxotroph, and the deletion of the MSBB gene, making its LPS less immunostimulatory, it has been attenuated. The strain has gone through phase 1 clinical trials and has a demonstrated safety profile as well. We first repurposed YS1646 for a prophylactic vaccine. And through prime boost vaccination with oral and intramuscular doses, we were able to reduce worm and egg burdens by over 80%, reaching up to 90% protection. We also observed a mixed Th1, Th2 immunity in both humoral and cellular responses, and as well as reductions in egg granuloma size, which is important for pathology, and an increase in the percentage of morphologically abnormal eggs, suggesting that this may have transmission interruption potential. So with the success of our prophylactic vaccine studies, we wanted to see if this vaccine had a dual purpose. It could be also used therapeutically to address the needs of the 250 million people previously mentioned, which are currently afflicted by the disease. And to test this, we infected 6 to 8 week old black 6 female mice for a period of either 2 months or 4 months leading up to our multimodal vaccination schedule, which consisted of 3 doses of oral doses of 1 times 10 to the 9 CFUs of Salmonella on days 1, 3, and 5, and a simultaneous dose of 20 micrograms of recombinant Cathepsin B given intramuscularly on day 1. Following vaccination, we followed the mice for a period of either 4 weeks or 8 weeks, where we then sacrificed them and assessed parasite burden as well as serum for different antibody responses and also spleens. We first looked at the parasite burden at 4 weeks, which relates to graphs A, B, and C, where we looked at relative worm reduction, relative reduction of eggs per gram of liver, and reduction of eggs per gram of intestine, where we saw 4 weeks post-vaccination, parasite burden reductions ranging from 46 to 50%. When we then looked at parasite burden reductions at 8 weeks post-vaccination, we saw an increase in these relative reductions, going up to 63%, suggesting that there might be a long-term effect of the vaccine over time. We then wanted to look at the IgG responses in the serum, where we measured anti-Cathepsin B IgG for both our PBS control group and our experimental vaccination group. The black bars refer to the control and the gray bars will refer to the experimental group. And the diagonal red lines refer to post-vaccination compared to pre-vaccination, which is the solid bars. So for both the 2-month and the 4-month infection groups, post-vaccination, we saw an increase in the experimental control in the experimental group and the anti-Cathepsin B IgG titers, we saw no increase in the titers for our PBS group over time. We then looked at the ratio of the IgG1 versus IgG2C antibodies, where IgG1 antibodies refer to more Th2 skewed immunity, which is common to see in chronic schistosomiasis, and IgG2C refers to a more Th1 related immunity. So in the first graph on the left, we see a ratio skewing way more towards IgG1 subtype, which is what we expected to see for our PBS control and also for our vaccinated group pre-vaccination. But post-vaccination, we saw a significant decrease in this ratio, where we led to a more balanced response of IgG1 and IgG2C subtypes, suggesting that we are observing a more mixed Th1 and Th2 immunity profile. On the graph on the right, we are assessing the total IgE antibody titers pre and post vaccination for both our control and our experimental groups. And this is important because historically, when testing helminth vaccines, and namely the hookworm vaccine, there have been complications in clinical trials where researchers have observed an increase in allergic-like responses when vaccinating individuals who have had previous exposure to the antigen through natural infection. So we wanted to see if we saw a significant increase in our vaccinated group compared to our PBS control post-vaccination. And what we saw was a trend towards an increase for both PBS and our vaccine group, but no significant difference pre or post-vaccination for our recombinant salmonella vaccine group, and no difference as well over time for our PBS control group. We then took spleen supernatants, which we re-stimulated and collected the supernatants to assess the secretion levels of different various cytokines by multiplex ELISA. So first, for the Th1 cytokine secretion levels, we saw significant increases for IL-1 alpha, IL-1 beta, IL-6, interferon gamma, as well as TNF alpha, suggesting once again that we're seeing a rise in Th1 immunity and taking us away from the skewed Th2 responses, which we see in chronic schistosomiasis. We also observed a significant decrease in the IL-2 levels in our vaccinated mice, which is interesting because IL-2 is associated with chronic pathology of the disease. We then also went to measure Th2 and regulatory cytokine secretion levels, where we saw a significant increase in levels of IL-5 as well as IL-10 cytokines in our vaccinated group, which is good to see because we are observing a mixed once again, a mixed Th1 and Th2 immune profile, and we're not skewing too far towards Th1 immunity, which would be lethal uh, historically for mice. So having this increase in IL-5 and IL-10 suggests that we're achieving a balanced immune response profile and taking us away from the skewed Th2 immunity that we see in our PBS control group. Lastly, we then looked at chemokine secretion levels, where we saw a significant decrease in levels of CCL2, which is a chemoattractant for monocytes, macrophages, and dendritic cells, as well as CCL3, which is a chemoattractant for macrophages. And this was of particular interest because CCL2 and CCL3 have strongly been associated with pathology in both mice and humans with chronic disease. And we saw a increase in the levels of CCL5, which is a chemoattractant for T cells and eosinophils, which may serve to explain the increased secretion levels that we're observing for both Th1 and Th2 cytokines. Lastly, by H&E staining, we looked at the eggs in the liver by histopathology, where we saw a significant decrease in the egg granuloma size of the eggs in our vaccinated group compared to our previous control. And we saw an increase in the percentage of egg morphologically abnormal eggs. So this decrease in area and as well as coupled with the increase in the abnormality of the egg morphology helps to explain the decrease in pathology that we've observed through the expression of certain cytokines and confirms this decrease in pathology as well as a potential, once again, interruption of a potential transmission interruption capabilities of our vaccines. Sorry. Therefore, our vaccines may not only be affecting the mice, but may also be having long-term effects on the fecundity of the female worms and their egg-laying abilities, leading to eggs that are morphologically abnormal, and as well as less able to stimulate an immune response in the liver and create a strong strong granuloma around the egg. So just as a summary, we have on the left pane what we see in untreated schistosomiasis, where we see lots of worms and eggs in the small intestine mesenteric veins, where the worms reside, and the eggs will be transiting either to the intestinal lumen or be taken up into the liver, where we see barely any Th1 cytokine responses and strong Th2 cytokine responses, as well as markers for pathology and loss of tissue fibrosis caused by these granulomas in the liver. Whereas on the right here, when we vaccinate therapeutically, we see a rise of anti-Cathepsin B antibodies, which serve to target the worms which are found in the mesenteric veins of the small intestine, as well as a decrease in the number of eggs found both in the liver and in the intestine, as well as a decrease in the overall parasite burden and the fibrosis in the liver seen by the reduction of egg granuloma size, where we also saw an increase in Th1 cytokines as well as Th2 cytokines. So with that, I would like to thank both my supervisors, Dr. Momer and Dow and Dr. Brian Ward, as well as all the members of both labs. I would like to thank all of our funding agency partners, and as well as you for your attention. Thank you.